The process of DNA replication is a really important one in life sciences. When you're looking at this process, it's important that you remember that when you make new cells, each cell that you have has your whole DNA code in it. So by making new cells, you need to make sure that the new cells that are produced will also have the identical DNA code as the original cell had, like in mitosis when you need to repair damaged tissue. The same DNA needs to be present in both of the daughter cells that are produced from the process of mitosis. So we ensure this by undergoing a process called DNA replication that happens during interphase. This is one of these processes that you need to learn word for word and be able to describe it. So when learning this, it's best to try and actually practice writing it down, not just simply reading through it, but being able to see a diagram and write what is happening at each step. So the first process is the natural shape of DNA is a double helix. So it's this double-stranded molecule that is in a twisted shape. So it's like a twisted ladder. But this can be a bit convoluted to deal with and for bonds between nitrogenous bases to break. So what happens is the first step is the DNA double helix unwinds. This then forms the typical ladder shape that you can see, with one strand running on the left and the other strand running on the right. The next step is that the weak hydrogen bonds between these nitrogenous bases, so guanine here is on the one strand and cytosine is part of the other strand, and these hydrogen bonds between nitrogenous bases, the paired ones that are base pairing in a complementary fashion, adenine, thymine, thymine, adenine, guanine, cytosine, etc., these hydrogen bonds need to break so that these nitrogenous bases can separate from each other. So you went from having a double helix shape to a ladder shape because the double helix unwound itself. And the next step involves these weak hydrogen bonds between the complementary strands, because guanine is complementary to cytosine, adenine complementary to thymine. These hydrogen bonds will break to form two separate strands. Now, it's important here that you mention that two separate strands are formed because the next step involves each strand acting as a template. So this one is the first template, which will form a complementary strand with it, and this is the second template, which will form a complementary strand. The complementary strands are formed by free nucleotides from the nucleoplasm that come and join on with its complementary partner. So guanine will have a cytosine nucleotide come and join on, adenine will have a thymine one, thymine will have an adenine one, guanine will end up having a cytosine one, and so on and so forth. So for the sake of this video, I have simplified the structure of a nucleotide. It's important to remember that there would be a sugar molecule here and there would be a phosphate either below, above or below, depending on the strand. But then what does happen is that the sugar phosphate bonds form and then there is a bond across all of the nucleotides and they eventually form a new strand. And the same thing will happen on the left-hand side, where on the original half of the strand you have a cytosine, a guanine nucleotide will come and bond there. There's a thymine, so an adenine nucleotide. There's an adenine, so a thymine nucleotide. All of these nucleotides are sitting in the nucleoplasm, in the nucleus. So the entire second strand will now be formed on the original strand. And of course, new hydrogen bonds will form between each of the nucleotides of the corresponding complementary strands. So that will happen across both of the new strands. These weak hydrogen bonds will reform. Once all the complementary nucleotides have joined on to the original strand and the hydrogen bonds have all reformed, you now end up with two DNA molecules. So the double helix will reform, those DNA molecules will recoil or retwist up themselves, and you end up with two genetically identical DNA molecules. So very important to say that they are genetically identical. Now, each one of these molecules will consist of one original strand. If you remember correctly, this was an original strand and that was an original strand of DNA. And it will also have one new strand of DNA. So that was a new strand and that was a new strand. So if we show it here, there will be one original strand of DNA on this molecule and one original strand of DNA on that molecule. And the other strand will be a new strand that was produced through complementary base pairing. This whole process is controlled by enzymes. You do not need to know the names of any of these enzymes, but you can see very clearly that these molecules are completely identical. You have guanine, adenine, thymine, guanine, guanine, adenine, cytosine, guanine on the left there, and the same on this side, guanine, adenine, thymine, guanine, guanine, adenine, cytosine, guanine.